Hello everybody, having a good hair day today, so I decided to make another video. So with that in mind, we're going to discuss DNA, which stands for deoxyribonucleic acids. It's one of our four major organic macromolecules. Along with nucleic acids, there are carbohydrates, lipids, and proteins. So let's begin. Nucleic acids are found in the nucleus. That much should be obvious. Nucleic acid, nucleus. And essentially these are what makes you, you. So that's what makes them so fascinating. And we're not necessarily going to talk about how they serve as the instructions for living things. That'll be a topic for next semester, but we'll lay the foundation for that now. There's two types we're going to discuss in detail. There's many more nucleic acids out there, but the two in detail are going to be DNA and RNA. DNA, as we've already talked about, is deoxyribonucleic acid, whereas RNA is just ribonucleic acid. They're both composed of long chains of nucleotides. So these are the building blocks for nucleic acids. Imagine I gave you a bunch of Legos, and I said, hey, build a fort, or a castle, or a house, or an airport, or whatever. If I gave you a bunch of nucleotides, you could build DNA or RNA or any other type of nucleic acid. So that's what we mean when we say, hey, these things are polymers. Polymers are long chains made of repeating subunits. So the monomers for that polymer, or the building blocks per se, are called nucleotides. So again, look at the word association there. Nucleic acids are made of nucleotides and are found in the nucleus. Nucleotides themselves have smaller components, one of which is a sugar. The D in DNA is what denotes that sugar. There's deoxyribose is the name of that sugar. Remember from our carbohydrate unit, a sugar or a carb is anything that ends in OSC. Now that's not always the case, but it's a good rule of thumb. In addition to sugar, there's also phosphates. So we talked about phosphates a little bit with our ADP and ATP unit, but the phosphates here occur or they exist in the presence of or form of phosphodiester bonds that happen between the nucleotides, and that's what is going to give DNA that classic shape we'll discuss in a moment. And last but not least, the different part between our different nucleotides is the base. So let's go back to the beginning. There's nucleic acids. We're going to discuss DNA and RNA. These nucleic acids are made of smaller parts called nucleotides. Each nucleotide is made up of three components. There's a sugar, a phosphate, and a base. And here we see them. Here's our sugar group, our phosphate group, and our nitrogenous base. They're called nitrogenous bases because they're heavy in nitrogen. See the 1, 2, 3, 4, 5 nitrogen atoms here? So you might see them referred to as nitrogen base, nitrogenous base, or in this class we're just going to call them the bases. The circle there is the main difference between the two sugars in DNA and RNA. There's deoxyribose and ribose nucleic acids. We talked about that before. You'll see here, around my mouse, that the oxygen is present in RNA, whereas in DNA there's only a hydrogen. The oxygen is gone. So let's think about that word once again. Deoxyribose versus ribose sugar. So basically, it's ribose sugar that's been deoxygenated, or they've taken one oxygen out. So think deoxygen, deoxyribonucleic acid. So once again, if you're ever struggling with a vocabulary word or a concept of any kind, try breaking that word down, and it usually will provide clues as to the context or meaning. So this is something many of us have seen before, and there's many more differences than just these three, but these are three fundamental differences between DNA and its close molecular cousin, RNA. DNA is a double helix, all right? It's two strands, and the helix, think helicopter, right? Helix, it means it's a twisted or a spiral shape, whereas RNA is a single strand. There's the four bases we're going to talk about in a minute. Well, of those four, thymine is the fourth base in DNA, whereas uracil is the fourth base in RNA. DNA is contained in chromosomes and it stores that hereditary information. Again, heredity is something we'll talk about next semester, but for now, just know that that heritable info is passed from one generation to the next in those chromosomes, and within those chromosomes is where this DNA is found. Whereas RNA is that key role in the manufacture of proteins. It serves as the middleman, middlewoman, whatever, between DNA and the nucleus, and then the ribosomes that we learned on our second unit, which is the site of protein production. You can see this nice double-stranded spiraling DNA helix here. See the phosphates? The sugars are these things in the middle. And then here you notice that the bases pair with one another. You see A with T, G with C. Those are held together with hydrogen bonds. Again, something we discussed. We said hydrogen bonds, while weak, do play an important role in, our lives. And we'll talk about why more in a moment. Here's another picture of DNA and then its close molecular cousin RNA. Notice up here there's A's, T's, G's, and C's, whereas down here there's A's, G's, C's, and U. There's U instead of T, and that's where that uracil as opposed to thymine comes in. Notice the deoxyribose in DNA versus the ribose in RNA. And again, let's make note of these hydrogen bonds that are occurring between the base pairs in the double strands. So again, that double helix, that comes from that sugar phosphate backbone. So that's where these phosphodiester bonds, that's a strong covalent bond that occurs between the third carbon on one molecule and then the fifth carbon on another sugar molecule, and that ends up helping that twisted shape occur. And that twist is how this DNA gets to be wound tighter and tighter and tighter, and then it supercoils inside these chromosomes, and you can fit so much stuff in such a small area, the nucleus. So here's another picture out to the side. You see this at a lot of universities. They'll have fountains that are like spiraled with water coming down. I know SEMO has something, and I've seen it in other places too, but just a homage to the DNA molecule and how important it is. Once again, notice G goes to C and A goes to T. So let's wrap up here. The genetic information is determined by the order of bases, as in AATTGGC is something completely different than AATTGGG. So once again, we'll talk more about that in the semester. We're just trying to lay the foundation, the basics now, but that's how that DNA is going to be different between you and I and any other living thing. And that's what's important to remember, is that all living things, all of them, you, me, a bumblebee, a dandelion, a white rhino, everything out there has DNA. And that DNA is composed of the same 4 bases. Now, depending on the different combinations of that DNA, you can have wildly different life forms. And just look at us. Between myself and a lot of you guys, we're all humans, but there's lots of differences. How can that be if we all have the same 4 bases? And that's going to give you kind of the insight into the scale that we're talking about here, how much DNA is really there. Just like a word is determined by a sequence of letters. So ATE, EAT, same 3 letters in a different order has completely different connotations. There are those 4 bases again. Adenine goes to thymine, guanine goes to cytosine. So like we've said already, these bases are going to pair. So I would come up right now with a nice mnemonic device that's going to help you guys keep that straight in your mind. They always pair with these hydrogen bonds. For me, A equals T, G equals C. I like to say Aaron and Travis's grandma is cool, because she was, right? Maybe you guys could come up with something on your own. It's Halloween time right now, so maybe all teens like good candy, whatever. All right, as long as you have A with T and G with C. The sooner you can lock that down, the easier the rest of this semester and then the first month of next semester is going to be. Once again, you see the A's, T's, G's, and C's in the middle. They make the rungs of this ladder or the steps of this staircase, whereas here is the sugar phosphate backbones, and that's what causes it to twist. So the bases themselves, they're also categorized into 2 groups. There's purines and pyrimidines. Purines are adenine and guanine, whereas pyrimidines are going to be cytosine and thymine. And don't forget uracil. We're not really going to talk about RNA yet, but we're just kind of introducing you now. Whereas cytosine and thymine, notice the fundamental difference between these. Your purines have 2 rings, whereas your pyrimidines have 1. With that in mind, our cytosine-guanine pair forms of 1, 2, 3 hydrogen bonds, where our adenine-thymine pair forms of 1, 2 hydrogen bonds. So here's a nice wrap-up of everything going on within this DNA unit. All right, here's your sugars, your phosphates, and then your 4 bases. Once again, the sugar phosphate backbone is what causes that double helix shape to occur. Notice that there's single-ringed and double-ringed bases. There's 2 hydrogen bonds between A and T and 3 between G and C. Note that A always goes with T and G always goes with C. And Here's a nice little wrap-up. Last thing we'll discuss is this double helix shape, book by Watson and Crick. We'll talk about how they worked with Rosalind Franklin, Wilkins, and a few others to help discover that shape. And again, why is DNA important? Because of what makes you, you.